Hello students, welcome to this video where we are going to discuss biochemistry recall questions of INI CET November 2022 exam. I have collected the questions of morning as well as evening sessions. So let's get started. First question here which I am taking in this PPT is, this question on glycogen phosphorylase was asked that this enzyme is activated directly or indirectly by. When we have done the video on glycogen, we have done this rate limiting enzyme of glycogenolysis, that is glycogen phosphorylase. It has two forms, small b and small a. Small b is inactive and small a is active here. So how to convert b to a? We need phosphorylation of this enzyme. And also we know that catabolic enzymes are activated in phosphorylated state. So now very easy to learn the activators and inhibitors. Anything which is doing phosphorylation, that will be the activator. Anything which is doing dephosphorylation, that will be the inhibitor. Okay. So activators are cyclic AMP, which causes phosphorylation. Hormones like glucagon, epinephrine, norepinephrine, right? Then calcium and 5-AMP. All these are activators, right? Inhibitors are phosphatase, which will remove the phosphate. Then insulin, which causes dephosphorylation. And then product inhibition. What is the product of this pathway? Glucose and energy, right? So product, glucose, glucose 6-phosphate, energy ATP, that will be the inhibitor. And also learn that fructose 1-phosphate is also the inhibitor here. And this is very important for hereditary fructose intolerance, which is very important topic. So do revise this topic for your upcoming NEET exam, right? So see the options. Options are calcium, which is activator, glucose 6-phosphate inhibitor, insulin inhibitor, glucose inhibitor. So what is the answer? Answer is calcium. Calcium will join with calmodulin and it directly activates the enzyme without phosphorylation, right? The mechanism of calcium calmodulin complex is direct activation of enzyme without phosphorylation. Coming to next question, question number 2 here of this PPT. Burnt sugar smell of urine is due to the defect of. Burnt sugar smell is occurring in MSUD, maple syrup urine disease. The name of disease itself tells you that there is burnt sugar like odor. Maple syrup, sweet kind of odor. Okay. So this is branch chain keto acid dehydrogenase, or also the name of this enzyme is branch chain keto acid decarboxylase. Okay. There is mental retardation in this disease, right? And this is defect in the catabolism of branch chain amino acids. Branch chain amino acids are valine, leucine and isoleucine, right? So what answer is D here. Let's look at other options. Phenylalanine hydroxylase. This enzyme is deficient in phenylketonuria. And what is the odor of body odor in phenylketonuria? It is mousy or musty odor, right? Then isovaleric CoA dehydrogenase, it occurs in isovaleric acidemia. This is defect in the metabolism of leucine branch chain amino acid. Only one, that is leucine, right? These patients, isovaleric acidemia patients, have rancid, cheesy or sweaty feet odor, right? Then fumarate acetoacetate hydrolase. This is the enzyme deficient in tyrosinemia type 1. Tyrosinemia type 1 is fumarate acetoacetate hydrolase. So let's also do tyrosinemia type 2 and 3. So type 1 we did. In type 1, the enzyme is fumarate acetoacetate hydrolase. Easy to learn. First tyrosinemia, the enzyme starts from F. Okay. Then Type 2 tyrosinemia, there are two T in the name of the enzyme, that is tyrosine transaminase. Tyrosine transaminase, right? So type 2, so two T's here, okay? T for tyrosine, T for transaminase, right? Then in type 3, it is deficiency of PHPP hydroxylase. PHPP hydroxylase, right? Here, this is complete deficiency of PHPP hydroxylase. If there is partial deficiency of this enzyme, if there is partial deficiency of PHPP hydroxylase, then the disease is Hawkinsinuria. Hawkinsinuria, where patients have swimming pool-like odor from their bodies, right? So, this is done with this question. Coming to next question, the significance of HMP shunt are all except. For solving this question, you need to know the full reaction of HMP. When I have taken the video on HMP, I have cut short that what you have to learn, because HMP is a big pathway. You cannot cram whole of it. It's a cycle actually, okay? So the main thing is that you learn the full reaction of HMP or HMP cycle. Then it is easy to solve so many questions, right? So in HMP cycle, we take 3 glucose 6-phosphates, 3 glucose 6-phosphates taken plus 6 NADP taken, 6 NADP taken. So these are the substrates you can say. And after the reaction or cycle, what you get is, you get 2 molecules of glucose 6-phosphate, right? So 3 were taken, 2 we have again obtained. Then 1 molecule of glyceraldehyde 3-phosphate, 1 molecule of glyceraldehyde 3-phosphate, then 3 carbon dioxide and 6 NADPH, 6 NADPH, right? So this is the full reaction. And also in phase 1, from phase 1, we also get ribose 5-phosphate, which is required for nucleotide synthesis, right? So let's read the options now. Significance of HMP shunt are all except, okay? So first is, it is source of NADPH. Yes, that's the function of HMP, to give NADPH. And we get how many? We get 6 NADPH, right? Then option B, source of ribose 5-phosphate. Yes, during the phase 2, the first step is that we obtain ribose 5-phosphate. Okay, I'll show you on next page. Then it is source of acetyl-CoA. How it can be source of acetyl-CoA? Indirectly, because this glyceraldehyde 3-phosphate will enter into glycolysis, as it is the intermediate of glycolysis. Now, via glycolysis, pyruvate will be formed, and from pyruvate, via link reaction, acetyl-CoA will be formed. Okay, so this is also correct. Then option D, it utilizes ribose. No, it will utilize ribulose to synthesize ribose, right? So this is wrong and this is the answer. Okay, so let's see the next slide. Look here, this is oxidative reactions of phase 1 of HMP, right? So till here we have phase 1, right? So in phase 1, from glucose 6-phosphate, we are getting ribulose 5-phosphate. There are mainly 1, 2 and 3 steps. Okay, so here we are getting NADPH, here also we are getting NADPH. So 2 NADPH are obtained in phase 1 of HMP, that is also one of the MCQ. Then carbon dioxide also we are getting in phase 1. So here look that we are getting ribulose 5-phosphate at the end of phase 1. Now when phase 2 is starting, this ribulose gets converted to ribose. So ribulose is used, ribose is produced. So now coming to next question, question number 4th here. A young male with a history of dyslipidemia having ABCA1 mutation shows the following. See the image and tell the diagnosis. So look at the image. The image here shows enlarged tonsils which are yellowish color. right? And what is this disease? This is very typical case of Tangiers disease, in which there is mutation in ABCA1 gene. This gene codes a transporter or channel, that is ABCA1, which is present on peripheral cells, and it helps in the exit of cholesterol and phospholipids from the cell to the HDL. Okay, this will be even more clear on next slide diagram. Look here, this is a peripheral cell and this is ABCA1 channel, right? Through this channel, cholesterol and phospholipids, they come out of the cell and they are incorporated in HDL, and then HDL will take it to liver. Okay, so this way the cholesterol and phospholipids of peripheral cells are going to liver, right? Now, if there is a mutation in this gene, then what will happen? The cholesterol and phospholipids, they cannot escape the cell, so there will be fat deposition in this patient at various organs. Example, liver and spleen will be enlarged due to fat deposition, leading to hepatosplenomegaly in Tangiers disease, right? Also, lymph nodes are enlarged and throat is also enlarged because tonsils are enlarged here, right? And tonsils, when they have fat deposition, then this lead to orange-yellow discoloration. So, this is very typical of Tangiers disease, right? Also, Tangiers disease is known as hypo-alpha lipoproteinemia, because alpha lipoprotein is HDL. Alpha lipoprotein is HDL. HDL will be not properly formed in this case. So there is decreased HDL or decreased alpha lipoprotein, right? Now, another option A is Wolman's disease. Can you tell me what is typical of Wolman's disease? For Tangiers disease, the typical image is orange-yellow tonsils. But for Wolman's disease, the typical picture is calcification of adrenals. This picture will be used in your image-based question if Wolman disease question is asked. Wolman disease is also known as cholesterol ester storage disease. Cholesterol ester storage disease. Here the enzyme deficient is acid lipase. Okay. Let's quickly revise. In which disease acid maltase is deficient? Acid maltase is deficient in Pompey's disease. Okay, Pompey's disease is a glycogen storage disease. Okay, so let me tell you some important topics for your upcoming NEET exam. Wolman disease and sphingolipidosis do all the diseases nicely. The way I have taken in the video is very shortcut and only the high yielding portion, which is required for you to solve the questions easily. Then glycogen storage diseases are also very important. Okay, do revise them. Then next question, fifth question. Cystic fibrosis is caused by which of the following deletion? In cystic fibrosis, there is scarring of lung tissue. Scarring of lung tissue will be called fibrosis, right? Now, F for fibrosis, F for phenylalanine. One letter abbreviation of phenylalanine is F. Okay. So, F for fibrosis, F for phenylalanine. In this mutation, there is deletion of one codon, which leads to absence of this one amino acid in the protein, that is phenylalanine at position 508. Okay. So, what is the role of this cystic fibrosis gene? This gene makes a protein, that is CFTR. CFTR protein, full form is cystic fibrosis transmembrane regulator. Okay, so this protein helps in the movement of water and chloride in and out of the cells and helps in formation of mucus secretions or saliva or digestive secretions, right? And when this gene is normal, then the mucus is thin and freely flowing, right? Have a look at this image. This is normal CFTR channel, So the channel is throwing chloride ions out of the cell, right? So this leads to thin layer of mucus, which is normally required and very much useful. But in case of mutant CFTR channel, now this channel is closed. Okay, so there is impaired movement of water and chloride across the channel, and this leads to a thick layer of mucus which is sticky. This will lead to airway obstruction due to sticky thick mucus. This will lead to infection and inflammation, and finally leading to scarring of lungs. Okay, so answer to this question, this you have to cram only, that cystic fibrosis, 1 amino acid deletion occurs, phenylalanine at 508 position, right? Then next question number 6 here, Refsum's disease and Zellweger syndrome, both these diseases were asked and both are related to subcellular organelle peroxisomes. Okay, so before reading the two questions, let me tell you about peroxisomes. Very, very important. Peroxisomes, these are organelles where some pathways occur but no ATP is produced in these pathways. What is produced is H2O2 is produced in such pathways, right? Or you can say other way, that any pathway which produces H2O2 will be occurring in peroxisome, right? So peroxisome have mechanism to take care of this because they have catalase enzyme, and this enzyme will destroy the produced H2O2, right? And everything is safe. Now, which are these two pathways mainly? One is alpha oxidation of fatty acid, another is oxidation of very long chain fatty acids. Okay. Now this alpha oxidation, I will write here. It occurs for branch chain fatty acids like phytanic acid. Okay. One of the enzymes required for this oxidation is phytanic acid hydroxylase. Now, when this enzyme is deficient, then this is Refsum's disease. Okay. So Refsum's disease is defect in alpha oxidation of branch chain fatty acids, right? So our answer here is B option. Let's look at other options. Option A, beta oxidation of branch chain fatty acids. No, beta oxidation mainly occur for palmitic or stearic acid, which are saturated and they are even chain fatty acids. Okay, not for branch chain. Beta oxidation cannot occur for branch chain fatty acids. That's why body has mechanism of alpha oxidation, right? Then option C says omega oxidation of branch chain fatty acids. No, omega oxidation is a type of oxidation where dicarboxylic acids are formed. The omega carbon is oxidized. Dicarboxylic acids are formed, right? Then option D, alpha oxidation of dicarboxylic acids. No, for dicarboxylic acids, alpha oxidation is not there. I already told you, alpha oxidation is for branch chain fatty acids, right? Now, there is one disease where dicarboxylic acidosis occurs. That is MCAD deficiency, which is also very, very important topic. Do revise this, MCAD deficiency. In MCAD deficiency, there is non-ketotic hypoglycemia. Okay. So, as we are doing this paper, recall questions of INI CET, we are revising some frequently asked topics, obviously, and we are also taking care that which topics more we have to revise for our upcoming NEET exam. Okay. So, that should be the intention when you are doing this paper. The intention should not be only to know the questions or the answers, right? So, next is Zellweger syndrome. I told you this Zellweger syndrome is also for peroxisomes. So, it is caused by defect of which following organelle? Answer is peroxisome. Now, what occurs in Zellweger syndrome? In Zellweger syndrome, there are empty peroxisomes. So, there are no enzymes in peroxisomes. Okay. When no enzymes means alpha oxidation is also not occurring. and oxidation of very long chain fatty acid is also not occurring. So in Zellweger syndrome, both phytanic acid and very long chain fatty acid will accumulate, right? And also note down that this Zellweger syndrome is most severe peroxisomal biogenesis disorder, PBD, peroxisomal biogenesis disorder, right? Questions are frequently asked on Refsum's disease and Zellweger syndrome, okay? Then our next question is on COVID virus, about the diagnosis. So question is, SARS CoV-2 diagnosis is done by all except. We all know that this virus is single-stranded RNA virus. So we have to detect for diagnosis, we have to detect the genome, which is RNA, or we can detect the antigen, right? So for antigen antibody reactions, it is immuno word is used. So immunoassay can be used, right? Then RT-PCR is reverse transcriptase PCR. So here the starting material is RNA. RNA is converted to DNA by reverse transcriptase, and then PCR can be done, okay? Then real-time PCR is for quantification of DNA. It is also called quantitative analysis or quantitative PCR, right? Now, blotting, southern blotting cannot be done for this because southern blotting is for DNA, right? And the genome here is RNA, right? What is northern blotting for? Northern blotting detects RNA and western blotting detects antigen or antibody, right? The next question, selenocysteine, 21st amino acid, very, very important topic. Do revise this whole topic. Here the question is asked that it is present in which enzyme? Selenocysteine is present in glutathione peroxidase. It is required for glutathione peroxidase, for thyroidoxine reductase, and iodothyronine deiodinase. So you have to cram these three enzymes: glutathione peroxidase, thyroidoxine reductase, and iodothyronine deiodinase. Okay. Also, you should remember the codon of selenocysteine. It is UGA, and it is formed, this amino acid is formed by co-translational modification. It is not post-translational modification, right? The next question number 10th, 2,3-BPG. We know that 2,3-BPG is formed in RL shunt, Rappaport-Lieberring shunt. So the question is, it causes unloading of oxygen to tissues because, okay. So we know that when 2,3-BPG is there, then it helps in release of oxygen from hemoglobin A at the tissue level, okay. So that hemoglobin give its oxygen to the tissues, right? Now this is formed in RL shunt, and we did the energetics of RL shunt in glycolysis topic, that there is no ATP formation in RL shunt. The purpose of RL shunt is to produce 2,3-BPG and not energy. So any option where ATP is written, that will be wrong, okay. Neither decrease or increase ATP production, right? Then option C is, it binds to the central cavity of hemoglobin surrounded by histidine and lysine and favors salt bridge formation. Yes, this is correct. You will clearly see this in the next slide. So look here, this is 2,3-BPG structure. There's too much of negative charge in it, right? There is too much of negative charge in it, right? Now look here, this is alpha chain, this is another alpha chain of hemoglobin. These are two beta chains, okay, 2 alpha, 2 beta, adult hemoglobin. Now in the central cavity here, 2,3-BPG is present, which has negative charge, and the hemoglobin has lots of histidine and lysine, that is positively charged amino acids. So there is high positive charge here. So due to interaction of these two opposite charges, there is formation of a salt bridge, right? So this is salt bridge formation, okay, salt bridge formation, and this structure of hemoglobin is called TOT structure, and TOT structure helps in deoxygenation, okay, deoxygenation. On the other hand, other structure is relaxed state of hemoglobin, relaxed state which helps in oxygenation and it happens at the level of lungs when hemoglobin needs to pick oxygen, right? Here 2,3 BPG levels are decreased. Okay. Now this was your answer, that it binds to the central cavity of hemoglobin. We just saw, surrounded by histidine and lysine, positively charged amino acids, and favors a salt bridge formation, right? Then option D says it inhibits TOD structure. No, it increases the formation of TOD structure. So answer here is C. Then next question, image-based question. Identify the left curve. This question is asking to identify this left green curve where the right curve is of adult hemoglobin, right? We all know that hemoglobin curve is sigmoidal, oxygen dissociation curve, sigmoidal, and this curve is of myoglobin. Myoglobin's curve is rectangular hyperbola, right? Myoglobin's curve is rectangular hyperbola, right? Fetal hemoglobin and carbon monoxide poisoning also shift the curve to left, but they are having sigmoidal shape of the graph, right? And acidosis will shift the curve to right side. Okay, so let's revise this. Oxygen dissociation curve, right side shifting reasons, acidosis, increased temperature, increased 2,3 BPG, right? On the other hand, left side curve will be because of alkalosis, fetal hemoglobin, carbon monoxide poisoning, and methemoglobinemia, right? So do learn this. This is very, very important. The next question, question number 12 here. True about sirtuins are all except. What are sirtuins? These are silent mating type information regulatory proteins. So these are proteins. These proteins help in delaying the aging process, right? How they delay the aging process? Because they are useful in DNA repair. They are responsible for DNA repair mechanisms, and they are doing it by base excision repair. Okay, so they delay aging and they improve glucose metabolism also. So they are very good proteins, regulatory proteins. Now let's read the options. Option A, increase insulin sensitivity and support glucose metabolism. Yes, it is correct. It is a histone deacetylase. Yes, it is histone deacetylase. I told you when there is histone deacetylation, so now histone will be positively charged because acetyl group has negative charge. Now acetyl group is removed, so histone has positive charge, DNA has negative charge. So these positive and negative charges will tightly bind with each other and it will lead to chromatin condensation, right? So this is also correct. And this thing is responsible for DNA repair mechanism, right? Then option C, it increases lifespan. Yes, it increases the lifespan because it delays aging. Option D, it is stimulated by niacinamide. No, this is wrong. It is inhibited by niacinamide. You will understand this on my next slide. Look here. This sirtuins have ADP ribosyl transferase enzymatic activity also. For this enzyme, the substrate is NADE and the product is nicotinamide or niacinamide. Okay, nicotinamide or niacinamide is one and the same thing. Now, nicotinamide inhibits, it will inhibit, any product will inhibit the enzyme. So, nicotinamide will inhibit, right? So, this was our answer, option D. It is stimulated by niacinamide. No, it is inhibited, right? Then question number 13. Write sequence of steps in the conversion of cholesterol to testosterone. This is a difficult question which was asked in exam. For this, you have to learn this big pathway of conversion of cholesterol to androgens, progesterone and mineralocorticoids, corticoids, right? So, the answer here is this option A, which I am showing here. 4 enzymes, desmolase, dehydrogenase, hydroxylase and lyase in a sequence. So, let's do this pathway. So, this is cholesterol. From cholesterol, testosterone is formed, right? So, the first enzyme is 20-22 desmolase, with the help of which cholesterol will be converted to pregnenolone, right? Then 3-beta hydroxysteroid dehydrogenase and progesterone is formed. Then 17-alpha hydroxylase and 17-hydroxyprogesterone is formed. Then 17-20 lyase, androstenedione is formed, and then finally testosterone is formed. So do cram this pathway, right? And also look this diagram to understand the number of carbons. Cholesterol has 27 carbons. There is loss of 6 carbons through many reactions, and we get pregnenolone, which is 21 carbons, which will further form progesterone, that is also having 21 carbons. Progesterone will make glucocorticoids and mineralocorticoids. So all these are 21 carbons. Now progesterone will be converted to androgens, which are 19 carbons, and then estrogens, which are 18 carbons. So this also can be an MCQ, that testosterone has how many carbons? It is 19. Estradiol has how many carbons? 18, right? Then next question, fructosamine, which is used for assessing long-term glycemic control in a person with hemolysis, is a glycated product of. See, in a patient with hemolysis, HbA1c cannot be done. HbA1c will be falsely decreased here. Falsely decreased because there is damage of RBCs, so hemoglobin, so we have lost hemoglobin also, right? So look here. So glucose, when attached to some proteins, these are glycated proteins. Glucose can be attached to albumin, called glycated albumin. And a term fructosamine is given to this category. Any glycated protein, especially albumin, that is fructosamine. Okay. Then what is HbA1c? When in RBCs, hemoglobin is attached to glucose, right? So this is HbA1c. But in hemolysis patient, we cannot do this. So we need fructosamine, and fructosamine is a glycated product of albumin. Answer is albumin, not fructose, not glycosaminoglycan, right? Be careful. So you can also have a look at this picture. So albumin joins with glucose. This is glycated albumin. In RBCs, hemoglobin joins with glucose. This is glycated hemoglobin, HbA1c, right? Images have a great impact on our mind. So whenever you do lots and lots of images while you're studying, that will help you a lot, using lots and lots of colors, right? Then we'll also learn falsely high value of HbA1c or falsely low value. When do we get this? So especially learn in iron deficiency anemia, there's falsely high value. In hemolysis, reticulocytosis, there is falsely low value, right? Then coming to next question, which of the following tissues are dependent on insulin for glucose uptake? So we have already done this, that GLUT4 is dependent on insulin, and this all is happening in fed state, and the peripheral tissues, muscles and adipose tissue are taking up glucose through GLUT4 in the presence of insulin, right? So this we learned from this table of GLUTs, which we did in our separate video on GLUTs, right? You know that I have made all the videos as short topics, like GLUTs is a different topic, glycolysis is a different topic, so that when you are doing these videos, it is very easy for you to complete the video and feeling good that you have completed. Without any burden, you can do these short videos and you can revise them also, right? Let's quickly revise. GLUT1 and GLUT3 are in fasting and they are doing basal glucose transport. Then GLUT2 in fed state, and it is present in intestine, liver, pancreas and kidneys. When pancreas get glucose in fed state, then insulin is released. Insulin will activate next GLUT, that is GLUT4. So it is present on cardiac muscles, skeletal muscles and adipose tissue. Okay. GLUT5 is for fructose, right? Then next question, Hook tragedy is related to which of the following alcohol? Hook tragedy is related to methyl alcohol. What do you mean by hook? Country liquor can be categorized as spurious liquor or hook when the amount of methane added could be fatal and death occurs suddenly, right? And sudden death occurs. In newspapers and media, we frequently get such incidences happening because of the making of spurious alcohol here and there, right? Now, what is the biochemical basis here? Methanol, which is an alcohol, so it will be taken by enzyme alcohol dehydrogenase, making formaldehyde. This is aldehyde, so aldehyde dehydrogenase enzyme will work and will convert it to formic acid. This formaldehyde and formic acid are very dangerous, leading to metabolic acidosis, blindness and neurological damage, right? So this is the reason that methanol or methyl alcohol is very, very dangerous, right? Now, the competitive inhibitors, fomepizole and ethanol can be used in the treatment part, right? Now, next question number 17, very, very easy, which is true, right? We all know that between A and T nitrogenous bases, there are 2 hydrogen bonds. Between G and C, there are 3 hydrogen bonds. So, obviously, high temperature will be required for breaking GC bond, right? So, the correct answer here is to break AT bonds, low melting temperature is required, right? All the other options are wrong. The next question is an image-based question on a graph from enzyme kinetics. We did this topic nicely in our video of enzyme kinetics. Please do revise whole of that video and revise other graphs like Lineweaver-Burke plot and the rectangular hyperbola graph also, right? Now, this is very easy question. Enzyme inhibition, which type of enzyme inhibition is asked? So, this is inhibited portion. It is very easy. You have to see the 2 lines are joining at which point? So, here Km. They are joining at x-axis where we have Km and Km is same here. Km is same in case of non-competitive inhibition, right? In case of competitive inhibition, Vmax does not changes. Okay, Vmax is same and Km is increased. Okay, do revise this thing for all the 3 type of inhibitions. Then question number 19th, assertion and reasoning type question. Assertion is phenylketonuria is characterized by mousy odor. Yes, this is true statement. There is mousy odor in phenylketonuria because of phenyl acetate. Okay, phenylalanine is in excess, so its metabolic product phenyl acetate is formed that gives mousy musty odor, right? But here what reason is given? Tyrosine and norepinephrine are not formed in phenylketonuria. This line is true, but it is not the reason for the assertion given. Okay, how it is true? Because in phenylketonuria, there is defect in the formation of tyrosine from phenylalanine. Here there is a blockage and tyrosine forms norepinephrine and other catecholamines. So obviously, tyrosine is not formed, norepinephrine is also not formed in this case of phenylketonuria. So what is the answer? Both assertion and reason are true and reason is not the correct explanation for the assertion, right? Then last question here, image based question. This cell is given, what is this diagnosis? This is Gaucher's disease. Gaucher's disease has crumpled tissue paper like appearance of the cell. So this cell or this cell or this cell, this is crumpled tissue paper like. And which enzyme is deficient? It is beta-glucosyl ceramidase. Beta-glucosyl ceramidase, right? So this is it with this video. Thanks for watching and congratulations to those who have performed well in this INI-CET exam and best wishes to those who have full hopes on themselves that they will be doing their best for the upcoming NEET exam, right?